let's kick off. First of all, I would like to welcome 2 times Johan from XNK Therapeutics. The company is not listed today, but we don't know what happens in the future. But you have a very exciting cell discovery that you've been working on for a long time. And please go on and kick off. And then we take questions in the end. And the listeners are, of course, more than welcome to post them in the chat on the right side. Welcome, Johan and Johan.
Thank you very much. We come from XNK Therapeutics, and we are working with natural killer cells, also called as NK cells. And we are today a world-leading company in the field of autologous NK cells. Here is our disclaimer. Please read that carefully. As you know, and was pointed out, we are not a public traded company today. So my name is Johan Living. I've been working the last 15 years at Janssen, the last 5 years in global roles, and by training, I'm an engineer in physics. And with me today, I have Johan.
So I'm the other Johan Ascon. I am MD, specialist in hematology, internal medicine, spent 20 years at the Karolinska Hospital and academia, and then 14 years within pharma, initially at Janssen, in different positions in Nordic, European, global roles, and then the last 2 years at Oncopeptides, and I'm happy to be here.
So let's look at the company in brief. Well, we are built around the world-leading research at NK cells that are going on here at the Karolinska Institutet. It was here the functionality of these cells were discovered in the mid-1970s. At our core, we do have a platform technology that is both stable and versatile and allow us to develop cell therapies for different cancer indications. We started in multiple myeloma, a blood cancer, and we come quite far. We have completed a phase 1/2 clinical trial with good safety results and a promising efficacy. And this summer, we recruited our first patient into a randomized phase 2 trial where we combine our autologous natural killer cell therapy with Sanot's antibody-dependent, which is an anti-CD38 antibody currently on the market. If you look at the potential of this type of company, it's quite big. It's due to the relatively large pool of patients available, the price points that you can achieve, and of course the scalability of the platform technology itself. When we started working in this field 2012, very few other companies, researchers believed that the NK cells could be used as a therapeutic option. And the company progressed, and something happened in the year 2018-2019. There were coming out clinical studies showing that the NK cells could actually work as a cell therapy against cancer. And then the whole market really took off. And today you can see a new deal basically every month, a major deal. And the big companies are also awakened and seeing, well, there is something here. The natural killer cells could be the next generation of cell therapies. If you can look at our part of the market, we're working with autologous NK cells, which means we're using your own natural killer cells and restore the capacity. We are currently working in multiple myeloma, and we are in phase 2. We're not alone. Biohaven, a US company, is in phase 1. If you look at the other companies, you see something interesting. They are target different kinds of cancer indications. Lung cancer, colorectal cancer, breast cancer. There is no reason to believe that our platform technology should work only in multiple myeloma, but we can scale this to other cancer indications. And this is a proof of concept that other companies have started at different points in their clinical development. If we take a deeper look at our company, we were founded back in 2012. We have today 3 patent families. The first is around our platform technology. The second is around the treatment of multiple myeloma. And the third is a combination therapy where we combine our autologous NK cells with a high-dose antiviral treatment. From the regulatory pathway, we have today an ODD status in multiple myeloma, both with the FDA and EMA. From the clinical development, we said we completed a phase 1/2 trial showing good safety profile, promising efficacy, and with this summer entered a phase 2 combination trial together with Sanot's anti-CD38 antibody. From a company structure point of view, we scaled up quite heavily. We have a nice and good management, very competent in place. Johan as our Chief Medical Officer. We have Mikael Uhlin, our Chief Scientific Officer, and we are today announcing that Marcus Thor is coming in as our Chief Business Officer. We're also quite well funded. We raised 64 million during the spring in April, and we can now accelerate at a very, very good pace. We are working with natural killer cells, NK cells. These are part of your body's primary immune system. They circulate the body and they're looking for cells that they would not believe belong to your own body. They can be infected with viruses, they can be transformed into cancer cells. And when a NK cell is finding such a cell, it will start its killing machinery. However, when you get cancer, the cancer somehow try to escape this mechanism. And our company is built on a very simple idea. Can we restore your own natural killer cell to its former glory? Can we activate them? Can they become cytotoxic again? And while we do that, can we then grow them in large numbers so we actually get a cell therapy? That's the simple idea that this whole company is built around. I actually believe we're on the right place to doing this as well. As said, it was here in the mid-70s where the functionality of the natural killer cells were discovered. In our board of directors, we have Hans Gustav Ljunggren, who made some of these basic research and early groundbreaking discoveries. So being a Nordic Swedish company working this field, I feel quite proud of doing that. Let's look at our platform technology. It's quite simple. From the patient point of view, it will come in, donate 1 unit of blood. That's 450 milliliters, simple blood donation. The patient will then go home. The blood is sent to us. We'll freeze it and store it. When it's time for production, we thaw it and we put it into our expansion activation protocol. That takes roughly 20 days. After 20 days, we have restored the activity of the NK cells. They become cytotoxic again, and we grow them up in large numbers, and we freeze the end product. Since the company has been around for quite a long time, we have 10 years stability of our end product. So if needed, we can keep it in our fridge for a very long time. When this comes for the treatment of the patients, the cell therapy is sent to the clinic. The patient come in and we thaw the product bedside and infusions to the patient. The patient will stay for observation for a little while and then go home. So from a patient point of view, it's quite simple. And for us, we have a good logistic procedure. The good thing about being an autologous NK cell therapy is that we can give multiple dosages because we do not have to immunosuppress the patient. So in our clinical trial program, we give 3 dosages. If we look at what happened in our expansion and activation, when the natural killer cells are circulating the body in a patient that has cancer, they do not do much. They do not have any cytotoxic capacity. And as you can see from the picture in the middle, then you have day 1, which is on the bottom. They do not kill your own multiple myeloma cells. But then we start our expansion activation process. And after 5 days, they start to show some cytotoxic capacity. And after 20 days, they have quite a strong cytotoxic capacity. So we know they can kill off your own multiple myeloma cells. But you don't want these NK cells to do any harm. You don't want them to kill normal cells because then you get quite severe side effects. So if you take our expanded and activated natural killer cells and put them and show them normal cells, healthy cells, well, at day 0, they don't do a lot. But after 20 days, well, they still do not attack them. They recognize them as your own natural killer cells. And that's the beauty of our cell therapy. So if we look at the scalability then at our platform, we started in multiple myeloma. We come quite far. We are in phase 2. But if you look at the other companies in this field, they targeted other cancer indications. So today we are looking into a different variety of other indications. And we're looking for 2 things. First, we would like to find a situation where we have a relatively low tumor burden. The good thing about that is that at the end, it's going to be a numbers game. Every NK cell can just kill out a certain amount of target cells. So if you have a low tumor burden, you have a higher likelihood of success. And also, there are quite many antibodies out there on the market, like Sanot's anti-CD38 antibody, which uses the body's NK cells and its killing machinery. And if you can combine that, it's a good synergistic effect. So I'll leave to you to talk a little bit more about our clinical development program.
Right. So as you heard, our leading indication is multiple myeloma. And what is that? Well, it's a rare form of blood cancer that affects the plasma cells in the bone marrow. And it's a horrible disease, as you can see on the X-ray picture, with all these black holes in the skeleton. So the tumor can cause severe bone pain, fractures, and also fatigue due to anemia and renal insufficiency. So truly affecting the quality of life of affected patients. And ultimately, this is an incurable cancer. Despite huge progress in treatment, novel treatments over the last couple of decades, it still remains fatal. And even if the overall survival has improved from roughly median 3 years to 6-7 years in median, as I said, it still causes a lot of morbidity and ultimately mortality. So there is a need for additional treatment options. So where are we? As you heard, the ACP-001 was our first in-man study, completed and done, reported last year. We are opening up for the combination study, as you heard, and I will come back to this. Further preclinical developing is ongoing in smoldering myeloma. It's an asymptomatic earlier stage of myeloma, and also in elderly patients, those that are not eligible for a high-dose therapy with transplant, because the current setting where we are running the clinical trials are as consolidation after first autologous stem cell transplantation in newly diagnosed myeloma patients. So let's look a little bit closer to the first-in-man study. This enrolled and treated 6 patients with 3 infusions each, with escalating doses of our expanded activated NK cells. The primary objective was, of course, to look at safety and tolerability. If possible, we had a look at the effect on the multiple myeloma, and there were, in addition, a number of exploratory endpoints. So to the left starts with the important outcome from the study, the safety. There was no serious adverse event, which is quite rare for a study in multiple myeloma patients. We did see adverse events. The majority were of the mild, the grade 1, and also unlikely to be related to the infusion of the stem cells, according to the investigator. However, what we did see was shingles in the first 4 patients. So shingles, this is this activation of the herpes zoster virus, which is dorming, sleeping in your body. And this is very interesting, actually, because it has been reported that NK cells can attack those cells where the herpes zoster virus are dorming. So this, you could say, is a proof of efficacy of the infused NK cells. And the good thing is that it's treatable, and even more important, you can prevent it by antiviral medication, which was done in the next 2 patients who did not develop shingles. We did see some cases of upper respiratory infection, back pain, diarrhea, headache, paresthesia, and most of them were, as I said, not related to the treatment and are common in this population of patients. 4 of the 6 patients had measurable tumor cells before the infusion. And in all 4 of them, we could see a declining level of either the tumor traces in the blood, or perhaps even more important, in the bone marrow by a minimal residual disease measurement. With a median follow-up of 5 years, all patients are alive. 4 patients have progressed in the myeloma, making the median progression-free survival to 34 months. There were signs of NK cells activation, and One of those is the granzyme B, which is a protein that is secreted from activated natural killer cells. We measure this in the blood. And in the middle of the slide, you can see this sawtooth pattern with increased levels of granzyme B in the blood after each of the 3 infusions. Perhaps even more important, to the right, in the bone marrow, where the tumor cell resides, we could detect 4 weeks, so 1 month after the last infusion, increased levels of granzyme B compared to before infusion. So all this, we believe, indicates a good safety, early promising efficacy results, which needs to be confirmed, but also surrogate markers speaking about NK cell activation. To build on the combination with an antibody, as Johan earlier described, we need to look at the hypothesis here. And what is important is that one of the most important activity by which the monoclonal antibody works is called ADCC, which stands for antibody-dependent cell-mediated toxicity. And how it works is shown in the cartoon here. When you give the antibodies, which are directed towards a specific antigen on the tumor cells, in myeloma, this is mostly CD38. The antibody binds to the tumor cells, but then they need a killing machinery from the NK cells, which is the second picture here with the blue round cell, which then links to this activating NK cell receptor, the CD16. It links to the antibody, which is marking the cell, and that triggers the apoptosis, the killing machinery to kick off in the NK cells. So antibodies are dependent on NK cells to have the full potential of its anti-tumor effect. And our hypothesis is that by adding a high number of activated NK cells, this will cause some additional cell killing. And that is actually the hypothesis behind this phase 2 study, which recently enrolled the first 2 patients. And this is a collaboration with Sanofi, us, of course, and the Karolinska Institutet and the hospital. And in this study, again, it's enrolling patients with newly diagnosed multiple myeloma, and this treatment is given as consolidation after the first autologous stem cell transplantations. It will enroll 60 patients that will be randomized to 2 different arms, and 30 patients will receive the NK cells together with the antibody, and the other 30 will only receive the antibody. And the purpose is to look at the overall response, including minimal residual disease in the bone marrow. There are some additional classical secondary endpoint and exploratory endpoints. So all this we can produce in our current laboratory. And this unit is the production unit at the Karolinska Cancer Center at Vecura in Huddinge. But to be able to expand our clinical research program and to be independent, we need to set up our own GMP production of the NK cells. And that is actually underway. And the plan is to at least be able to provide 100 batches per year to be able to fulfill our clinical study plans.
So looking into the potential, we briefly mentioned that the potential of these companies are quite big. So looking at only multiple myeloma, the number of patients for our first indication as a consolidation after autologous stem cell transplantation, they are somewhere between 30,000 and 40,000 patients. The price points that you can achieve if you compare to CAR T therapies, well, these are all in hematological malignancies. They are somewhere between 3 and 450,000 US dollars, so quite high price points. If you want to compare to the available antibodies like Sarclisa or Darzalex that are on the market for treating multiple myeloma, they are on the slightly $100,000 plus. And this is looking only in multiple myeloma. And then if you look at the scalability potential of this platform technology and what we can do in other indications, the potential become quite big very quickly. So just to sum it up, XNK Therapeutics. We are built around the groundbreaking research in NK cells that have been going on here at the Karolinska Institutet since the mid-70s. At our core, We have a unique and very good platform technology that allow us to restore and activate the body's own natural killer cells again and use that as a cell therapy. We come quite far. We have completed a phase 1/2 clinical data, see good safety and a promising efficacy, and we just launched a randomized phase 2 clinical trial. And the potential, if you look at this kind of company, is very high. Thank you very much, and happy to take any question if there are any.
I can see there's one patient actually regarding many patients that will be tested in US and Europe in the combination study. And this is actually a single site here at the Karolinska University Hospital. They do have a large number of newly diagnosed patients every year, and they are serving close to 2 million people in this area. So it will not be open in US, and as I said, only at the Karolinska unit here at the Huddinge Hospital.
Thanks a lot, Johan and Johan, for your nice presentation. If we look into to the phase 2 study and you succeed, and as you mentioned, it covers 60 enrolled patients. Do you have enough GMP capacity to take it into the next stage as a phase 3, or do you have to build out? Maybe it's a little too early, but you mentioned you had around 100 batches a year in the plant production. If you could answer that question.
I can take that one. So a good question to start with. So the hypothesis is that we need to look at the data. We are also planning another phase 2 study with only the XNK cells, because that arm is lacking in the combination study. So then we could compare antibodies, XNK cells, or the combination. And that will be the foundation for the hypothesis for the phase 3 study. And with also, in turn, depending on the statistical assumption, build the size of the phase 3 study. But our thinking is that we will have the capacity in the new lab, because this 100 is sort of a minimum. We have some capacity to further expand the production to 100 plus. So we believe that will be sufficient for our own phase 2 and for the forthcoming phase 3 study. So yes.
And also including smaller phase 1 trials for other indications that we are interested in pushing.
Yeah, and this phase 2 study, just to understand you right, it's third line patients you are treating there.
No. It's first line patients. We are going in first line in transplant eligible patients. They will receive the standard induction therapy, the auto transplant, and then after the auto transplant, as a consolidation, when the patient has been debulked, have a smaller tumor burden, we believe the cell works the best. Have the greatest opportunity to further, reduce the tumor cells in the body, and hopefully even down to MRD negativity, which is a very strong prognostic marker for overall survival in this cohort of patients.
And the reason for going so early, of course, the side effect profile that we see.
And I think that there are several arguments for moving into early. We harvest the material for the production of the cells before the patient have started the anti-cancer therapy. In that way, we believe we have the best opportunity to have really strong growth and expansion and activation of the cells. Because later down the line, when the patient has received a lot of cancer therapy, the cells might be less sort of active and more exhausted, so to speak.
Yeah, for sure. So what you address here is like, for instance, chemotherapy that kills everything. So if you get in earlier than that, you can get more cells from the patient's own body.
Exactly. We can get a better cell product, and when we give the cells, the patient have already been what we call debulked, so have had some received the treatment and reduced the tumor burden to a lower degree, and the cells have a greater chance to outnumber them and also be effective.
Yes. Then there's a question about what does this actually cost? And now you're not a public company, so you can, I don't know if you can answer that question. The phase 2 study.
we're not gonna answer that. It is an investigator-initiated trial, and we're only providing our NK cell therapy. So for us, it's a relatively low cost, and it's fully funded. So we are fine.
You took down the ongoing study, or
yeah.
Okay.
Yeah. And then there's a question here from the audience: when are they able to buy your stocks? That's a good question.
We are evaluating. I mean, currently, I have to come from the ground. We are well financed today. We have the capacity. We have the capital needed for really, grow the company. But we're looking into all different types of financial options in the future. So we haven't ruled out anything.
No, good. And then just to address in the end, because now we covered several areas within immuno and cell therapy today, and could you combine? You mentioned you could combine with the antibodies, but can you combine NK cells with CAR T cells? Because we have two very interesting companies in Sweden, and this collaboration that Agneta addressed in the beginning. If you could comment on that, it's probably a stupid question, but just to let the audience know.
On the opposite, I think it's a brilliant question, actually. And the short answer is we don't know. It has to be done. Theoretically, there is some possibility, but it all depends on the timing, etc., and the sequencing, how you give this. But the CAR Ts and the NK cells are working via different mechanisms. So I think from a theoretic point of view, there's a possibility to combine them. And that is the way the body is working today, by nature, combine the different modes of immune systems. And why shouldn't we do it for patients?
Yeah. Well, thanks a lot, Johan and Johan. Really interesting presentation. There's no further questions. And I thank all the participants and the audience for listening into this seminar on cell therapy presented by H.C. Andersen Capital. And we hope we can meet the companies in the future. I know about Elysea and Hydrogen, but hopefully also XNK Therapeutics. Thanks a lot and have a very nice day.
Thank you.
Thank you.
Welcome.
Bye-bye.